Welcome, dear viewer, to another exciting session of SHS1 Integrated Science. I am Edward Santiakufu, your presenter. In this lesson, we'll be focusing on cell structures, or what we refer to as organelles. In our previous lesson, we looked at viruses and cells. We studied the types of cells and some cell organelles. Today, or in this lesson, we shall continue with the rest of the organelles. Before that, let us look at the objectives for this lesson. By the end of this lesson, you will be able to describe the structure and functions of the following organelles: cell wall, cytoplasm, nucleus, endoplasmic reticulum, Golgi apparatus, mitochondrion, lysosome, vacuole, chloroplast, and centrole. We begin with the first one, cell wall. This is a structure that surrounds the cell membrane. The cell wall is found in bacteria, fungi, and plants. In bacteria, the cell wall is made of protein and fats. The cell wall of fungi is made up of chitin, whilst that of higher plants is made up of cellulose. Cellulose has very large molecules which form fibers. These fibers make the wall strong and supportive, but at the same time porous. The porous nature of the walls make it permeable to water and dissolved particles, allowing them to pass through relatively easily. Some plant cells have cytoplasmic link which crosses the walls. These links of cytoplasm are known as plasmodesmata. Now let's look at the functions of the cell wall. It protects and supports the cell to resist bursting during turgidity. And by turgidity, we mean the enlarged form of the cell as a result of water getting into the cell. It provides shape to the cell, especially plant cells. It is easily permeable to gases, water, and other substances. Another structure found with the cell has to do with the cytoplasm. The cytoplasm is a transparent watery fluid which is about 90% water, within which can be found various organelles. It houses the organelles. It is the site for many chemical reactions. It is the site for building up materials such as proteins and carbohydrates. It is the site where energy is supplied for cell activities. It is the site where food materials such as glycogen in animals and starch in plants are stored. Another structure you find in a cell is the nucleus. The nucleus is spherical or ovoid in shape and the largest cell organelle. It is enclosed by a double membrane called nuclear envelope or nuclear membrane. The nuclear membrane is perforated by nuclear pores, which controls the exchange of materials between the nucleus and the cytoplasm. The nucleus contains chromatin materials that form chromosome when the cell is about to divide. Chromatin contains DNA, the molecule of inheritance which controls the activity of the cell. The nucleus also contains one or more nucleoli, or nucleolus in the singular form, which manufactures ribosomes. The nucleus and the cytoplasm constitute the protoplasm of the cell. The only cell without a nucleus is the red blood cell in animals. Now let's look at the function of the nucleus. The nucleus controls the life activities of the cell. It contains the hereditary material, that is DNA, which is passed on from parents to offspring. The nucleus contains nucleolus, which synthesizes RNA for protein synthesis. It initiates cell division during the life process like growth and reproduction. Now let's move on to endoplasmic reticulum. It is a network of minute sheets and tubes bounded by a membrane. It is found throughout the cytoplasm and connects to the nucleus and the cell membrane. There are two types of endoplasmic reticulum. That is the rough endoplasmic reticulum and the smooth endoplasmic reticulum. The rough endoplasmic reticulum has ribosomes on its outer surface, and that gives it the rough appearance. However, the smooth endoplasmic reticulum does not have ribosomes on its outer surface. The endoplasmic reticulum divides the internal part of the cell into compartments so that chemical reactions occurring in the cell do not interfere with one another. Now let's look at the function of the endoplasmic reticulum. The rough endoplasmic reticulum synthesize and transport proteins. The rough endoplasmic reticulum serves as the platform that supports the ribosomes. The smooth endoplasmic reticulum synthesize and transport lipids, that is fats and oils. The endoplasmic reticulum provides the surface for chemical reactions to occur. Let's turn our attention now to the Golgi apparatus, or what you call the Golgi bodies. Golgi apparatus consists of a stack of flattened membranous sacs. This organelle, which is named after the biologist who first reported of it, is present in all cells. However, it is prominent in cells that are metabolically active. The stack of flattened membranous sacs have clusters of vesicles at their edges. The vesicles are small bodies in the cell containing secretory materials. The Golgi apparatus is numerous in secreting cells, such as cells of the salivary glands and the pancreas. Now let's look at the function of the Golgi apparatus. It is responsible for the production and repair of the cell membrane. It synthesizes complex carbohydrates. It is involved in the packaging and secretion of proteins and complex carbohydrates. We now move on to the mitochondrion. It is a sausage-shaped structure bounded by a double membrane. The number of mitochondria in a cell depends on the energy requirement of the cell, or where the cell is found in the organism. This is because mitochondria is responsible for the synthesis of energy in the form of ATP, which is adenosine triphosphate. For example, mitochondria are numerous in the muscle cells, the middle piece of the sperm cell, cells at the base of the wings of insects, and at the base of the quill feathers of the wings of birds. All these structures require much energy for their activities. Mitochondria contain enzymes which takes part in the respiratory process. It is therefore referred to as the powerhouse of the cell. Now let's look at the function of the mitochondria. It is a site for energy production in the form of ATP, which is adenosine triphosphate. Another organelle found in the cell is the lysosomes. Lysosomes originate either from Golgi apparatus or directly from the endoplasmic reticulum. They are bounded by a single membrane and contain concentrated mixture of hydrolytic digestive enzymes. If these digestive enzymes were not kept enclosed in a sac, they would attack the other cell organelles. They are numerous in animal cells than in plant cells. The enzymes they contain are used in the dissolution and digestion of redundant structures or damaged organelles from within or outside the cell. For example, when a cell dies, its own lysosomes release enzymes that digest the remains of the cell. This process is known as autolysis. What it means is that the redundant organelle will be broken down or digested by the lysosomes. So if any part of the cell needs to be destroyed, it is the work of the lysosomes to produce their enzymes to break that part of the cell up. Now let's look at the function of the lysosomes. It destroys itself when it is worn out, and therefore it is referred to as the suicide bag of the cell. It destroys redundant structures or organelles within the cell. Now, another organelle you find in the cell is the vacuoles. Vacuoles are fluid-filled cavities bounded by a single membrane called tonoplast, that is in the case of the plant cell. It occupies over 80% of the cell volume. The plant vacuole is filled with a liquid known as cell sap, an aqueous solution of dissolved food materials, iron, waste products, and pigments. The vacuole of animal cells are usually very much smaller and less permanent. Small vacuoles are often called vesicles. So what it means that some animal cells even do not have vacuoles at all. They may contain engulfed solids or liquids, such as food vacuoles or contractile vacuoles in the case of amoeba. Now let's look at the functions of the vacuole. Vacuoles provide support for herbaceous plants when they are turgid. They store food in plant cells. They provide the water balance, what we refer to as osmoregulation, in animal cells, for example, in the amoeba, paramecium, and some other unicellular organisms. Another organelle we need to look at has to do with the chloroplast. It is a large disc-shaped organelle bounded by a double membrane. They are found only in green parts in plants. They contain the green pigment, that is the chlorophyll, which absorbs the sun's energy for photosynthesis. We focus on the function of the chloroplast. It is the site for photosynthesis due to the presence of chlorophyll. What this means is that the chloroplast is not an organelle found in the animal cell. It is only peculiar to the plant cell. Ribosome is another organelle we need to look at. Ribosomes are minute structures which are composed of RNA and proteins in roughly equal numbers. Some are free in the cytoplasm, whilst others are bound to the outer surface of the rough endoplasmic reticulum. Let's look at their functions. They are the site for protein synthesis. And by that we mean it is within the ribosomes that proteins are manufactured. Centrioles are also one of the organelles within a cell. Centrioles are two hollow cylindrical bodies lying at right angles to each other beside the nucleus. They occur in animal cells and not in plant cells. They separate and move to the opposite poles at early stages of cell division. It is also believed to be responsible for the formation of spindle fibers. Now at this point, let's recap all the issues we have looked at in this lesson. We looked at the various cell organelles in both plant and animal cells and their functions. I hope you have enjoyed today's lesson, but I want you to continue to refer to the questions. Solve those questions, refer to the notes, and if there are any difficulties, do not hesitate to let me know. I will see you soon.